Hello, my name is Lily Arbizu. I am a team member of Connections Official, a company that specializes in genealogy research and research for the certification of Sephardic lineage for Portuguese citizenship. Today, I will review information about genetic genealogy as it relates to Y-DNA and Y-DNA testing. The objectives of the presentation include a review of the fundamentals of DNA biology, inheritance of Y-DNA testing methods such as short tandem repeat and single nucleotide polymorphism testing, as well as the potential significance of Y-DNA test results in genealogical research. On this image, the large spheric structures represent human cells, whereas the small rod-like structures inside the nucleus of each cell correspond to chromosomes, which are the carriers of genetic information in most organisms. As you can see here, each chromosome consists of one very long string of DNA that is tightly coiled, and the DNA in each chromosome contains thousands of genes. Now, under an electron microscope, the DNA looks like a twisted ladder that is held by what appear to be the rungs of the ladder. These are four nucleotides or nitrogenous base pairs named adenine, thymine, guanine, and cytosine. Different organisms have a different number of chromosomes. In humans, each cell normally contains 23 pairs of chromosomes. There are 22 pairs called autosomal chromosomes or autosomes, which are classified from 1 to 22 on the basis of size. For example, chromosome 1 is the largest, and it is over 3 times larger than chromosome 22. Autosomal chromosomes carry genetic information such as a person's temperament and physical appearance. For example, the color of your eyes, the color and texture of your hair, your facial features, height, and body type, and even specific skills such as your athletic ability or your artistic ability. Autosomal chromosomes look and function in the same way for biological males and females. This means an autosome may determine the color of your skin and whether you have dimples or not, regardless of whether you are a biological male or female. However, the 23rd pair determines the biology of sex, and they are different between biological males and females. Females have 2 copies of the X chromosome, while males have 1 X chromosome and 1 Y chromosome. So a child receives 1 chromosome from each pair a parent has. In other words, a child gets half of the chromosomes from the mother and half from the father. This explains why children inherit some traits from their mother and other traits from their father. So the children of a couple may inherit the Y and the X chromosomes from their mother and father as follows. Without exception, the father will pass down his Y chromosome exclusively to his male children. Likewise, he will pass his only X chromosome invariably to all his female children. Since the mother carries 2 X chromosomes, one from her mother and one from her father, she may pass down her paternal chromosome X or she may pass down her maternal chromosome X to either her male or female children. The Y chromosome is passed down from fathers to sons virtually unchanged, that is, without recombination. Therefore, a man's Y DNA is nearly identical to his great-grandfathers, great-grandfathers, fathers, and so on throughout the generations over hundreds or even thousands of years. For this reason, the Y chromosome can be used to discover in-depth paternal lineage, because again, it is not recombined in every generation. What are some of the potential benefits of Y-DNA testing? As just mentioned, the Y chromosome remains virtually unchanged for centuries and perhaps millennia. Therefore, Y-DNA testing may allow a person to determine the ethnic or the geographic origin of the paternal line. For example, if a man's paternal line had African or Native American roots, the segments of Y DNA related to his lineage will be preserved throughout dozens of generations. Additionally, Y DNA testing may help to narrow down genetic matches on a specific paternal line. For example, Y DNA testing may provide guidance to male adoptees searching for clues about their biological paternal relatives. Another potential benefit is that since the family surname tends to follow the patrilineal line in many cultures, generally there is a strong correlation between the family name and the Y chromosome. Once a potential surname is identified, a person can research the origin of the surname, whether German, Russian, or Welsh. Y DNA testing is not perfect, and there are some caveats around it. For example, Y DNA testing could reveal misattributed parentage. In other words, the test results may show a completely different family name connection, or it may show unexpected genetic matches. This might be a concern if an adoptee is unaware about the adoption or if a child is the product of infidelity. Also, a limitation with having an unchanging Y chromosome is that it cannot clearly determine who, among male relatives in past generations, is a direct ancestor. The reason is that male descendants of a paternal line share the same Y DNA, regardless of how long ago they lived. The relationship may involve father and sons, male cousins, or uncles and nephews. Yet, Y DNA testing provides valuable information that can be complemented with traditional research based on family records. An obvious caveat is that since the Y chromosome is only inherited by men, women cannot take a Y DNA test. However, they can test a male sibling for Y DNA to help break down any patrilineal brick walls in genealogical research. It is helpful to understand how the Y chromosome is inherited through one's family tree. So the easiest way to determine a potential test taker is to draw or print out a family tree for your target ancestor and highlight all men descending from that ancestor who would have inherited that man's Y chromosome. There might be a broken paternal line to the target ancestor. In other words, if a father only has daughters and not sons, then the Y chromosome inheritance stops with that father. This occurrence is referred to as a line daughter out. Now, there are two different types of Y DNA testing available: short tandem repeat and single nucleotide polymorphism testing. STR testing is most commonly used to begin Y DNA research. STR markers are short regions located along the Y chromosome, and the test looks at these markers to find sequences of DNA units that repeat a variable number of times. Those unique sequences are passed down through generations, and they can help determine a genetic relationship between two test takers. So, as mentioned initially, the genetic code is made up of 4 nucleotides or nitrogenous base molecules: adenine, guanine, thymine, and cytosine, AGTC. If the sequence GATTA is repeated 3 times at a certain position on the Y chromosome, then the DNA result for that marker position is 3. In the example below, the results from testing of 4 participants is shown at just 1 marker position of the Y chromosome. And male 1 has 8 repeats of the sequence, while participants 2 and 3 have 9 repeats, and male 4 has 10 repeats. Regarding Y DNA SNP testing, the test looks for variations from the norm in individual positions on the chromosome. Sometimes this is the result of mutations or aberrant behaviors in the gene, such as errors of translation or replication. Once a SNP mutation occurs, it will typically be passed down through subsequent generations, and it is unlikely to revert back to the default value. As such, SNP testing can be used to understand a genetic family tree, which is commonly referred to as haplotree. So, how are Y-DNA testing and test results relevant to genealogical research? Comparing the values of the same markers for two or more men can determine if they are a genetic match. And the more values that match, the more likely these men are to be related. Also, the more the markers tested, the greater the accuracy in determining if test takers are related to each other. Y-DNA STR testing can provide a haplogroup estimate, and Y-DNA SNP testing can be more accurate and provide a definite haplogroup determination. A Y-DNA haplogroup starts with a main branch, which over time divides over and over into increasingly narrower subgroups. So haplogroups may help identify the specific ancestral DNA tree from which the Y chromosome of a test taker originates. Subgroups are defined by one or more SNP mutations, and each unique mutation will determine the ancestral and ethnic origins of the haplogroup. Thank you for joining me today. To request a free evaluation of your Sephardic lineage for Portuguese citizenship, you may contact us at www.connectionsofficial.com, and you can fill out our questionnaire, and a free evaluation would be given to you in relation to your lineage, your Sephardic lineage. Thank you again for joining me today.